I'm Nicole.
And I'm Stacy. And we want to show you a simple experiment that demonstrates DNA extraction from a strawberry. Do strawberries have DNA?
Why, yes, Stacy, it does. DNA is found in the nucleus of a cell, and it contains the genetic material within it. Every single living thing of a cell has a nucleus, which contains that genetic material, and that it contains the instructions that produce other strawberries.
Wow. I thought only humans have DNA.
Oh, you learn something every day.
Yes, you do. All right, so a couple of fun facts about strawberries. The first one is that we found out while researching that there's approximately 9 feet of DNA in every single strawberry. And the second fun fact is that strawberry cells are actually octoploids, so they actually have 8 sets of chromosomes versus the 2 sets that we normally see in diploid cells. Wow. A lot of DNA. All right, so you're going to need 2 strawberries and a plastic bag. And you're going to just put your strawberries in the plastic bag and begin to smush them. It's probably easier to have strawberries that are room temperature versus really cold strawberries. We're just going to mush them up really well, trying to get this solid solution to a more liquid state. And then we're going to actually add a lysis solution to it. And lysis just means to break open. Nicole's doing a great job of squishing the strawberries. Thank you. You can actually smell them too when you're smushing it. Can you smell them?
And it just makes you want to eat them.
I know. I didn't notice that earlier.
Okay, I think I've squished them enough.
All right. So now Nicole is going to make our lysis solution. And for that, you're going to need 1/2 cup of water.
Which is already in it.
Exactly. And you're going to add 2 teaspoons of Dawn soap. We've heard from other experiments that Dawn soap is actually one of the better soaps to use. Not sure why. Maybe it's because it's more sudsy. And you're going to need about 3/4 of a teaspoon of salt. Stirring those 3 things together really well. And just to tell you a little reasoning behind each ingredient, soap actually works to get in between the water and the fats that surround the cell membranes. And the salt is actually going to help to punch holes in the membrane, helping us to get to the DNA a little easier. Nicole's just going to add that lysis solution to the smashed strawberries. And we're going to make sure that we really get the solution really well mixed together, and also allowing for time for each one of those ingredients to begin working on the strawberries. And remember, your first go around of doing an experiment is not successful. Don't get deterred from it. Just try it again. All right. And then when it's completely mixed well, you're going to want to funnel the mixture, and we're using a coffee filter. because you want to separate the liquid portion of the strawberry mixture from the actual solid strawberry. Maybe we should try to do like just a little bit more so that there's, because you want to, in a moment, we're going to actually add alcohol to this mixture. And the purpose of the alcohol is actually going to be to pull the DNA from this liquid, because the DNA is not soluble in alcohol. So you want to make sure that you add the same amount of alcohol solution to your strawberry liquid. And the alcohol actually it helps to form our precipitant. And a precipitant is just the solid, which is our DNA. And this way you're going to be able to touch it if you want. I'm going to let Nicole touch it if she wants. I'm okay with just seeing DNA. And then try to make sure to not mix the alcohol to your strawberry liquid. You want to take your glass and tilt it. Yep, to the side. Maybe just a little bit more. And then just very, very slowly pour that alcohol toward, okay, and stop so that you can see it go into the top. Just letting it move across it. You see it, how it's totally separated now? I don't know if you can see this on camera, but the white alcohol, I mean, it's clear. It's totally clear. Yeah, I'm going to keep going. And don't be so worried about if you're exact or not. See, it's not. We need to go a little bit more. Just the important thing is to go slowly, giving the alcohol an opportunity to just sit on top. Okay? You see it, Nicole? See the little webbing?
I see the little... Yeah.
It looks like little spider webs, literally. And it'll take just a couple of seconds. This really is cool. Yeah? Okay. And then we're going to try... The tricky thing is really to catch some of that webbing, but while Nicole is trying to do that, we hope that our experiment will inspire you to do your own experiment. There are tons of experiments that you can do at home. You can use solutions like distilled water, regular tap water, salt water, glycerin, and each one of those solutions has a different effect on things. And for example, each one of those solutions you can put into a small container, add a gummy bear to it, and then if you'll notice, each one of those solutions has a different impact on that gummy bear. So we hope that this will encourage you to want to do your own experiment to find out exactly why you get the results of the gummy bears. So you'll have a better understanding of what a hypertonic, hypotonic, and what an isotonic solution will do.
And this is Winston. What gene do you think is missing from Winston compared to his fellow species with hair?
I have to do an experiment or some research to find out. Bye.